Hello. Now I can hear me. So we're almost at the weekend here, and we figured we'd accelerate the pace a little bit. So we have a really packed program for this afternoon. This session is going to be a session of data showcase, and that means we're going to go through several sets of resources for data analysis and data science in neuroimaging, very broadly defined. With several speakers presenting several different resources. So we're going to try to go through this kind of marathon of different resources being thrown at you, try to see what things fit into things that you're interested in exploring maybe next week. The idea is to really give you some resources that would maybe match the ideas that you have for next week. So having said that, and in the interest of staying roughly on time here, let's get started with Melanie Gansman, who will present about PET imaging.
Thank you so much, Ariel. Thanks everyone for still being attentive. I know it's been a long week for all of you guys, but a very exciting week. I speak of experience because in 2017 I also attended NeuroHack Week, and this kind of got me started on all of what I'm going to present to you now. Because back then I was, I would say, enrolled into the BITS fold by back then Chris Gorgolewski, who was the main driver at that time point of BITS. So I'm going to tell you a little bit about open science efforts that are ongoing for PET imaging, just to make sure, because I know a lot of people are not working with PET. By PET, I mean not animals, but I mean, of course, post-emission tomography. So I'm going to tell you a little bit about the different standards we have developed in the last couple of years, and also what kind of pipelines for processing we're at the moment still developing to make things easier and more reproducible. So I'll quickly tell you a little bit about the PET bits format, just to let you know the slides are already up in the GitHub repo, and I converted to PDFs, and all the links should be working, so you don't need to worry about not being able to go to the relevant repositories and so on. All the links should be working. So I'll show you first what actually is the PET bits format. So we'll take yet another look at bits, and then in a quick look at the Jupyter notebook, I show you actually hands-on how to convert regular imaging data set that comes off of the scanner using a PET to bits converter that was developed by the Open PET project. And there will be a much longer version available in the bit starter kit as soon as that is merged. And then I'll also very quickly, only in the interest of time, show you how it would be possible to run a single PET analysis with the processing pipeline that's also developed by the Open PET project and is based on PET Surfer, which is a PET processing extension of FreeSurfer. But let's get going. So a lot of you guys, I think, when you're thinking about PET, probably think about oncology and the biggest application that we have in the clinic. And that's, of course, FDG imaging. While FDG imaging is super useful in the clinic and a very prominent tool, there's so much more to PET. With PET, we can do also dynamic imaging, and that really allows us to visualize and quantitatively measure the biological function. So, you know, with fMRI, we could do one thing. With PET, we can dive into yet another world. And in terms of standardization, the PET field actually already back in 2007 published a paper regarding consensus nomenclature for in vivo imaging, because at that point, it wasn't even clear, I would say, what the underlying terms were when people were publishing with respect to, for example, binding of radioligands to receptors. So all of this was already standardized in 2007. Now, this was followed up just here in 2020 by a paper that focused very much on all the ongoing efforts with respect to reproducibility and the replication crisis, but also with respect to the standardization efforts that were being made, for example, through the COBITAS report in the MRI world. So in 2020, a bunch of people from around the world came together, and we basically tried to make guidelines for the content and format of the PET brain data in publications, but also in archives. So this paper was very much focused on when we report experiments in publications, what needs to be there. And at the same time, we kind of got also a consensus on when we are sharing data in order to be able to repeat experiments, what kinds of things need to be recorded in the data. And this was then easily followed up by saying, okay, now we have these guidelines. So let's then make an extension of the brain imaging data structure for PET. And a lot of you guys have heard about BITS already. I'm going to not touch on it very long here in the interest of time. We'll have a breakout session next week on Wednesday at 3:30, where Ariel, me, and Taylor that are attending NeuroHackademy will introduce you to, what is BITS about, a little bit of history, what is like being a part of it, either of the steering group or also as a lead of an extension proposal or as a maintainer. So if you're interested in BITS, then come and join us then. So looking at the PET bin standard, it's as you have already been introduced before, for example, by Dora and others with regards to EEG or IEG. It's a way of standardizing our experiments and the data that come with it. And the most important thing when we're doing positron emission tomography imaging is time. And the reason for this is that we're working with radioactive ligands that are injected into humans, animals, in the thing you want to image. And of course, then we're dealing with radioactive decay, and that means our whole experiment is inherently time dependent. And therefore, it's very important in your overall experimental description to have a time zero defined. This could be either the injection of the tracer. For example, when we have, let's say, a carbon-11 based tracer, carbon-11 has a half-life of 20 minutes. This means then basically you inject and then you do the image right away. Then, for example, our time zero is usually injection of tracer, which often also equals our scan start. But then in other types of tracers, for example, for 18-based tracers like FDG, they're injected and then actually maybe there's a waiting time of 45 minutes or an hour until the actual imaging is performed. And then it's a question, what should our time zero be, since everything needs to be, of course, decay corrected appropriately. So what is also a little bit different in PET than from the other modalities is that the framing of the actual image frames is also often time dependent and varies across time. So due to the radioactive decay and that you have less and less signal as you're imaging, the frames usually get longer towards the end, especially if you're looking at a tracer that is fast decaying, for example, carbon-11, a base tracer. That's, for example, usually not the case for FDG. There they maybe take 5-minute frames. But in any ways, it's very important to know that frames can vary in time, and it's not just a simple image. So that, of course, needs to be appropriately recorded. And then we also in PET have an additional metadata in a lot of the experiments, definitely not all of them, but in a lot of experiments, especially when we're looking at drug development and maybe also looking at occupancy and so on, we actually are taking blood samples. And those blood samples are, of course, also in need to be corrected to the correct time of injection or scan start, and also need to be related to each other, because some of them are often done with an automatic sampler and some of them might be done manually at the same time. So how does the PET bit standard then look like on disk? You've seen this type of folder several times before. You have your study folder. Then inside, you basically have your description of the data in a JSON file. Again, you've seen probably those .json files several times, but those are just simply structured text files. So it's just a text file with some curly brackets. And here we can see, for example, what kind of data set this is, who the authors were, their relevant papers and DOIs and funding that can be acknowledged. Then as a second thing, we have a subfolder that is called PET under our subject and under the first session. And in this PET folder, we have also a JSON file, again, structured text file, and this accompanies our PET image. And in the structured PET the text file, we are then given the scanner details, but also very important details, just as the tracer name. So what was injected, what was the actual radionuclide, how much radioactivity was injected, and so on. And then further on is, of course, the actual imaging data. Here you can see an example of actual image data, frames of the imaging data in blue. And then from this, you could also extract time activity curves. In some cases, for example, you don't have an actual image, but maybe you only have the time activity curves already extracted. That is also some cases when you're doing region-based imaging. And then further on, there could be some blood samples. The blood samples, again, could be drawn manually or with an autosampler. There is, again, a structured text file describing the machinery that was used for blood sampling. And then together with it, there's also a TSV, so that's a tabular text file that has all the actual measurements of the blood in it. And finally, most often nowadays, we also have an MRI that's either quite at the same time or close to the PET scan. And this is then stored in regular its MRI format as well and also available. So this is actually a relatively, I would say, simple extension of the MRI way of storing things, with the little curl that we needed to be very careful in PET to make sure that we have a lot more information present in our PET JSON file, because it all needs to relate appropriately to the radioactive decay, but also it needs to relate, for example, spatially correctly to the MRI. Side note here is, for example, usually people doing MRI or Fmri, they don't really care if their images are corrected for gradient nonlinearities. They're just used like this. Well, for PET, this really matters, because when you have a PET image, it's not distorted by gradient nonlinearities. And if you then take an MR image that hasn't been corrected for gradient nonlinearities, they actually don't overlap. The space, the physical space that's shown in two images is slightly different. So those are details that are very important when you're then doing combined PET and MRI analysis. And the PET bit standard has been published here online now earlier this year. And if you want to have a look at how it works, how to get a pull request through, you can also still see the old pull request in the BITS GitHub. Now, for data conversion, what we wanted to build is a tool, a quite, let's say, an easy tool to use. So you can take the raw data off your scanner and just convert it to PET bits. And in order to do this, we wrote this little library called Pet2Bits, or better, the OpenOR PET people did, most notably Anthony Galassi, one of also the BITS maintainers now, and Cyril Pernet, together with Martin Nørgaard. And what they looked at is actually to build up a phantom data set collection, where they tried to collect different phantom data from all around the world, from all types of different PET scanners. And with those, they tried to make a library that can convert it. One of the, I would say, again, peculiarities of PET is that we have different imaging formats depending on what PET scanner you're on. While in MRI, DICOM is, I would say, almost exclusively used, at least when doing regular structural and functional imaging. In PET, for example, Siemens scanners, the Siemens high resolution research tomograph, it spits out this proprietary ECAD format that also needs to be handled, and where no documentation can be found at all, not even when trying to dig back into Siemens archives. So that's why we said, okay, let's make sure to have a representative phantom dataset collection. And I'm going to show you very quickly how that looks like in the Jupyter Lab. So you should see my Jupyter Lab screen now. Can I just get a thumbs up for that or a hand up? Yes, cool. I ran this already, so you have this Python notebook available to you on the Jupyter Hub. Basically, you just need to import some relevant packages. I also imported here GDown because we have a very small data set that we are sharing with you, hosted on GitHub, that is just two examples, an ECAD and a DICOM example, source data and the converted data. Then we can just with pip install our Pet2Bits, and then it's actually quite simple. You just download our example file. There's a little bit of looking into it and unzipping the file. Then you can actually see this is a raw data set. So you have, this is a folder where you see a bits folder. Basically, there's some code converted to it. There's a data set description, and there's a source folder. So that's the actual raw data that comes off the scanner. There's one that's the Siemens biograph that actually spits out DICOM. And then further on down, there's also a Siemens HRT that actually spits out this .v or ECAD format. And those two are then converted. There are examples also of having them converted in Nifty and JSON files following the PET-bit standard. And basically, once you have that example data set loaded down, and you've already installed the Python package PyPet2Bits, you just run our nice extension to DCM2Nix, which is DCM2Nix for PET, and it will just convert everything. Just as DCM2X, it spits out a lot of warnings, like, for example, it says, this thing is not found. As long as most of the necessary things and the required things are there, this should be running without problems. In case you would run into problems, you could always rerun that command and actually add arguments to it. This is in detail also documented in the Head2Bits repo, but also in the Bit Starter Kit. So you can then almost easily add certain values that you want to override in your JSON file. Additionally, also, you can take the JSON file just off the disk and double click it and open it and edit text file. But obviously we like it better if you actually try to ensure that you have everything inside some kind of parameters on disk. And we are also developing tools for this. That is everything with respect to conversion. But now let's get quickly to the processing. So our whole motivation with Head2Bits was that what we want to do is do something like this. Make it easier to build an atlas, for example, across a lot of pet data sets, and do this also fully automatically. So our underlying tool that we've now started working with and that we basically had already used for our serotonin atlas is PetSurfer. PetSurfer was developed by a free surfer developer, Doug Grieve, and published already back in 2014. And it basically can do some of the regular, I would say, pre-processing steps that are necessary for pet analysis, but also it can do some of the simple genetic models. This is being extended now as we are speaking. And I'm not going to go into this, I just have it there so you know. So PetSurfer pre-processing, there's a lot of stuff to do with the pet image. For example, you need the time-weighted average in order to register it with the MRI. And then once you have it registered to the MRI, you segment the MRI and extract region-wise values. But you also maybe want to extract voxel-wise values. And then you can also do this whole exercise on the surface, and you can also do it only subcortically in order to avoid partial volume effects. And then finally, once you have some basically time activity curves, either per voxel, per vertex, or per region, you do what we call genetic modeling, which is a type of differential equation modeling that actually will extract your finalized images, those binding potential images that we know when, or at least look at dynamic pet. And all of this has been developed. It's in the Jupyter notebook right now. So in the Jupyter notebook, the second part, what we did is we made a slightly updated version of our existing open NeuroPet data set that is also linked there. You can just get it done with wget. Then you can download PyPetSurfer and just run an example Py. You cannot do it on the hub right now because we couldn't get FreeSurfer to install and run on the hub, and FreeSurfer is necessary because we're doing the registrations with FreeSurfer for it to run. But in principle, once you're at home, you're on a computer where you have FreeSurfer installed or server, you should be able to go through the notebook and download the data set that we linked in there and then run a pet analysis. And then I want to just thank the team. This is the OpenNeuroPet team involved. We have the National Institutes of Mental Health, the Martino Center in Stanford, and then our site in Europe, NRU Rigshospital, and we're funded by the Novo Nordisk Foundation and the Brain Initiative Grant. Thank you for that, and I hope now I'm short enough that we have some time for questions.
So yeah, we do have a couple of minutes for questions. There was one question in Slack about whether Pet2Bits also works for SPECT images.
We haven't tried, but we could easily test it. Let me open Slack. So ping me later on. So Arthur, yes, I see you. It should be relatively straightforward. We also work with SPECT on site. So in that sense, this would be a natural extension to try. I think it mostly depends on SPECT scanner manufacturers, because the idiosyncrasies come from the manufacturer side for conversion.
Okay, other questions here in the room or on Zoom? If not, the next presentation is Manuela. Oh, there you are. Okay, Manuela Dorado Novoa from ADDI. I will share my screen here now and her presentation. You can take off your mask, yeah. Then we start the presentation.
Hi, everyone. Can you hear me fine? Okay, so my name is Manuela. I am an intern with ADDI, and today I'm going to be giving you an introduction to ADDI and the AD Workbench. So what is ADDI? So ADDI stands for Alzheimer's Disease Data Initiative, and we're a 501c3 MRO, Gates Ventures funded, and we're dedicated to fundamentally transforming the way that Alzheimer's disease is researched and shared with other collaborators as well in the scientific community. We're doing this through different ways. One of them is through a free data sharing platform where not only researchers, but scientists in the community can share their data, but also access high quality data that is not only easily accessible, but also very comprehensive, which I remember in one of the talks that one of the professors gave earlier this week. They were talking about what if all data was suddenly accessible, would that be useful enough or not? And certainly we tried to at least gather very high quality data that is very easily comprehensible, that has descriptors. So if you are not a data sharing, a data provider, but you want to access data, you can definitely be assured that we're going to offer very high quality data. Also, we offer data science tools, such as a virtual machine that has a very high computing power. I know that this is a very common limitation for a lot of researchers around the world. I'm from Colombia, and for me to do scientific research in a university in Colombia, it's very hard to find a lab that will have computers with a lot of computing power to do analysis. Also through funding opportunities and global collaborations. So today we're mainly going to be talking about the workspace where you can do the data analysis. We're not going to be talking too much about the data we have on the site because that's not too relevant today. Although if you have questions about that, you can let me know. So what is the workspace and why is it important? So the workspace is where you actually do all your work. You can actually analyze your data there, curate it, visualize it. You can share it with other team members. You can also access your virtual machine there, and you can store your data if you want. This is kind of what it looks like. You have access to our data sets, which are very easily accessible. We have data from genomics to sequencing to imaging to clinical. Also there you can access your workspace, and over there in the AD Connect, you can access the forum where you can ask questions. So this is again what the workspace looks like. You can request several workspaces if you have several teams. And within the workspace, you can do several things. So the first one is access your virtual machine, which is my favorite tool, and we'll talk about that later. The virtual machine has about 5 teras of storage that you can use. If you need more, you can always request more. In the virtual machine, you can access pre-installed softwares, or you can install your own. Also, you can access all the files that you have in your workspace, and you can share them with other team members. You can also explore mini apps. So mini apps are apps built with R, so you can visualize data. They're very easily, they're created very easily. If you already have one, you can upload it, or you can deploy it from our knowledge base, which has several mini apps that will help you analyze different types of data. We'll look into that later. You can also view data, edit data, store data, save data, download data, share with other team members. And we also have an R console as well as our Studio and Jupyter notebooks already pre-installed. And from there, you can access your files. So again, like I said, the workspace has up to 5 teras of data storage, but if you do need more, you can always ask for more. I would say one of the benefits of the workspace is that it's free. So that gives a huge advantage, especially to researchers around the world that don't have access to computing power, like I said earlier. Can I play this video? Do you know how I can play video? Oh, okay. Okay. Thank you. So this is just a very short example of some of the data analysis modules that we have. We have over 20 data analysis, and this is just one of the tools that we offer. It's very easily accessible. If you do already have data sets uploaded to your workspace, or if you have CSV files, you can just select them and then go to the data analysis modules, choose the type of analysis that you would like to run, whether statistical analysis, descriptive analysis, or maybe you want to run some visualization. You can always do that from here. And the cool thing about it is that if you don't know what something is, because working in science, sometimes you're asked to do plots or graphs or things that there's just so much stuff that you don't know. It will take you to the knowledge base where it will explain to you what each parameter is and what the recommendations are depending on the type of data that you have. And the cool thing is also that the graphs come out almost as if you're doing it in MATLAB. They're very nice. So this is some of the analysis that you can do. You can do statistical modeling, testing, descriptive statistics, data visualization. I think this looks better. Each model explains exactly what you can do with that. And again, in the little icon on the bottom left, you can always go into our knowledge base and see how that can help you and what you need. So another tool that you can access in our workspace is RStudio and Jupyter Notebooks, which are already pre-installed in your workspace. You don't need to install them. And they come with several packages. We'll look into that later, but if you also want to install your own packages, there's instructions on that and it's fairly easy. Here's a look at the mini apps that I was telling you about earlier. Here are some apps that we've created already. They're very easily built. From here, for example, I'm going to show you a mini app that I built later today that helps visualize DICOM images. Here. So here we're actually looking at MRI scans of a patient that's in his 70s that has clearly very enlarged lateral ventricles and a lesion in his right hemisphere, probably from a hematoma. But anyways, that's not important. What's important is the types of things that you can do with this. On our knowledge base, we also have different mini apps that if you like, you can deploy them into your workspace so you don't have to create them. The code is open source, so that comes in handy. I made a video for this, but I'm just gonna skip through it for the sake of time. But as you can see, there's several examples of, trying to change the quality here. But anyways, there are several examples of mini apps that will help you analyze different types of data like sequencing, or just run different types of analysis like linear regression, or run different tests. And this is my favorite tool, the one that I've used the most during my internship, which is the virtual desktop, and the one that I see the most usability because of the type of things that I've had to go through as a scientist back in my home country, and the type of barriers that we have in places where access to high computing is just not prevalent like it is here. So you can request a virtual desktop. Is free. You can request it for Linux or Windows environment. You can request multiple if you want. So if you have several teams, you can request more than one. The cool thing about the Linux, which is my favorite one, is that you can have more, I think up to 20 or 30 users at the same time. So if you're working on the same data, you can do it there, and you have like different desktops, even though you're using the same virtual machine. In the Windows, there's only one user at a time that can use it, but you can do the same things, whichever you feel more comfortable using. The standard virtual machine comes with up to 16 gigabytes of memory, But I understand that sometimes is not enough. It comes with 4 cores as well. But if you do need more, you can always request more. You can just reach out to the service desk and they'll provide more. And again, I would say that the fact that it's free, it's amazing. But if you want to use the files drive, which is where you can share with other collaborators, then you get 5 teras of storage. And these are some of the softwares that are already included with your virtual machine. I'm not going to go over them in detail, but these come pre-installed. If you want to install more, which I did because I like to do a lot of image analysis, so I install FreeSurfer, Cat12, SPM12, blah, blah. You can also do that. And this is just a short video of how you can access your own files in your virtual machine, assuming that you already have your data uploaded, which maybe I shouldn't skip this video, but you can see that it's very easy to upload your data in whichever format it is. And once it's saved, and you already have access to your virtual machine, if you don't, you can request it and it'll take no more than 48 hours to be deployed. Then you can easily access it from your virtual machine. Just like a regular computer. And this is an example of something I did. I don't know if I'm running out of time. Okay. This is an example of something you can do. So I don't know if you're familiar with FreeSurfer. You can install FreeSurfer. If you don't know how to install it in a virtual machine, you can always ask, and the team will help you install it. Right here, we're just doing analysis on T1 weighted structural MRI of cannabis users at baseline and 3 years follow-up. And this is just a very short video. Of course, this is not showing the pre-processing, but that's what I wanted to talk about today, the pre-processing. Any of you is familiar with how long it takes you to pre-process a subject in FreeSurfer? Yeah, how long? Yeah, if you're lucky. Yeah, so a lot of computers don't come with many cores. With FreeSurfer, you can run 1 subject per core that you have. If you have, say, 5 cores, you can run 5 subjects at the same time, parallel pre-processing. That means that if you have a data set of 100 subjects, you're going to have to wait about 8 hours per 5 subjects. That's going to take a long time. The cool thing about the virtual desktop is that even though it comes, the standard desktop comes with 4 cores, you can always ask for more. There's no problem with that. And that saves so much time, especially for researchers that simply just don't have access to this type of computing. And this is another example of something you can do. Right here, we're just doing analysis on ADNI data. I don't know if you're familiar with ADNI. It's just the Alzheimer's Disease Neuroimaging Initiative. And we're doing it on Cat12. Cat12 runs with SPM12, which is another neuroimaging software, and MATLAB. I know some people don't have access to MATLAB or don't have a license with MATLAB, and sometimes that's pricey. So Cat12 can run, they have a version that can run without a MATLAB license. But the cool thing about this is that similar problem, pre-processing takes a long time. Cat12 lasts less than FreeSurfer, it takes less time. However, if you don't have enough cores, you may not be able to pre-process all of your subjects in a timely manner. And here, this is just an example of 6 AD subjects and 6 control subjects. So it took me very little. I had 4 cores, 12 subjects. So that took me about, I would say Cat12 is about 1 hour per subject. And because I'm using parallel pre-processing, I was able to run 3 subjects in 1 hour. So a total of 4 hours for all my subjects. But in reality, the data set comes with hundreds of subjects. And I wanted just to leave it here because I haven't done that yet, but I did it with FreeSurfer and it took me just a couple of days. So that's amazing. So I'm sure that it will take me very little and I'll be able to see, of course, many more results with a lot more subjects that I'm not able to see here. Yeah, I actually went pretty fast, but that's it. It's there's so many more things in the Addi workbench that I would like to talk about, but for the sake of the NeuroHackademy, I just wanted to talk about the data analysis tools, but there's a lot more you can do. And I hope that if you do have questions, you let me know, and you're welcome to use it anytime. Thank you.
We do have time for a few questions here from Zoom or here in the auditorium. You have a question?
I'm guessing the Addi data are not in the platform. Do you think they will be at some stage, or the Addi data? Which data set are on the platform already?
So ADDI is about 2 years old. So because we're not very old, we don't have tons of data sets, but also we're being very careful as to the data sets that we want to collect. Right now, you can find data sets from the EPAD consortium. We have several longitudinal studies there as well. We have a couple of neuroimaging imaging data sets, but we want to make sure that we have enough softwares installed in our workbench so that people can analyze those data sets. So for example, the presentation that they just gave about PET images, it would be amazing to take a look at that and see if that's something that users can access with their PET data in our workbench. There's also a lot of data sets that include clinical data as well as sequencing data. We also have, I forget, we just added a few more data sets, but I didn't really prepare a presentation for that because I didn't think that would be the focus of this presentation. But you're welcome to create an account and see what we have.
There are a couple of questions in Slack. First of all, Patricia says, great initiative, and then asks if it's possible to store data in ADDI and analyze them on your own machine. So can you download data from there into your own machine, then analyze? Cool. Do you want to, if you can analyze them with MATLAB there or on your own, can you download the data down from ADDI?
Yes, there is some data that you can download. There is some data that you cannot. My understanding is that the EPAD data, you cannot download it. You can access it in the virtual machine, and you can process it there, analyze it, and then your results, you can download it, but you cannot download the data per se. You can analyze the data within the virtual machine with MATLAB, which is what I like to do. So I simply just install MATLAB there, and it's very easy. Yeah.
So just to verify that I understood the last one, you can use your own MATLAB license in the, yeah, okay, that's great. Okay, are there more questions? There is actually one more question from Patricia. So she asked quite a few here, and you can go and answer on Slack some of these, but thank you for all the questions. I didn't mean to say, but that's not good. And Caitlin is typing, I can see. So she might be typing answers. So maybe let's move on to the next speaker. Thank you, Manuela, for that presentation.
Hello, NeuroHackademy. Can everyone hear me? Yeah, good. Okay. Hi, my name is Adam Richie-Halford. I'm a postdoc at the Brain Development and Education Lab at Stanford, and previously a postdoc working here with Ariel. I'd like to talk to you today about the Healthy Brain Network, and specifically the preprocessed open diffusion derivatives from the Healthy Brain Network. It's an exciting data set, and I think some of you might find it useful during the hackathon. Okay, so most importantly, I'd like to thank my many collaborators, and I always try to put this up front in case they run out of time. I don't want to give them a short shrift. So all these fine people worked with me on this project, including about 114 people in this line. So I'll get to that in a little bit. I also should have mentioned I posted this website on Slack, and if you would like to follow along, you can go to that link there. All the gory details that I'm going to talk about today are in a preprint, which is in review at SciData. Okay, so I told you that I'm going to talk about the Healthy Brain Network. I should describe this data set first before talking about the diffusion derivatives. The Healthy Brain Network is an ongoing effort to collect clinical assessments and MRI, EEG from about 10,000 New York City area children at 4 sites. Staten Island, let's see, Rutgers, Cornell Brain Imaging Center, City University of New York, and Staten Island. Staten Island is a mobile 1.5T scanner. And it includes a broad range of behavioral, cognitive, lifestyle information as well. The imaging data is all openly available. You don't need a data usage agreement to access it. You don't need to pay anything. It's just on S3, on FCP Indy. And for further details about the original HBN study, you can see the data descriptor, which is linked there, or their helpful scientific data portal, which is linked there. Okay, so I mentioned the imaging data. You might be so excited already to get your fingers on this data set that you want to know, like, where is this? Where is this data? So it's stored on AWS S3 in that bucket there. And you can access using AWS CLI or your favorite SDK in Python or R or MATLAB. And within that bucket, you'll find a bunch of folders. There's an EEG folder with about 3,500 participants, and then a bunch of raw MRI. The bottom one there is the original HBN MRI folder, which about a year ago, that's all that existed. And the stuff that I'm going to describe to you now helped create those first 2 lines. And then there's a bunch of pre-processed MRI data so that you don't have to go through and wait the 8 to 16 hours per participant to pre-process. You can go to these folders. So inside of BIDS curated QSI prep for the diffusion derivatives, AFQ, I believe we had a tractometry talk earlier in the week, so some of you might know what that is. And then there are also some CPAC pre-processed derivatives for a smaller number of subjects. In addition to the imaging data, there's also a wealth of phenotypical data. Only age, sex, and handedness are openly available. If you'd like access to the other data, you need to sign a data usage agreement. But if you do, you get access to these clinician-administered case ads. And then depending on what the participants answer in case ads, they may be directed to diagnostic-specific assessments on like speech and language, intellectual disability, tic disorders, things like that. There's also the full Wyatt and Whisk for intelligence and learning. If you're just interested in language development in childhood and adolescence, there are a lot of assessments there, the SELF, the Tower, CTOP. There are also self-administered assessments on family structure, so on and so forth, and some novel data, which is kind of interesting that I haven't actually looked at, but apparently they collected the deciduous baby teeth, so you can go analyze those if you want. They did voice recordings, actigraphy. There's some links there if you would like to figure out how to download the phenotypical data. So great, I've got you all excited. Let's pretend this is a year ago. I've got you all excited about this data set and you want to use it. Well, there's a challenge. One is that the data is BIDS-ish, which is a nice way of saying not BIDS compliant. And that there was no publicly available pre-processed diffusion data, and also no quality measures along with this data. So that was kind of annoying. That's what we aimed to fix. So I'll give a brief overview of our approach. We started with the raw data. At the time, there were about 2,700 subjects in the raw data. We passed 2,600 of those through BIDS. And then of those, we passed 2,100 through QSI prep for pre-processing. And then we wanted to measure data quality. So we did sort of a hybrid QC approach where we selected 200 participants for expert QC, about 1,800 participants, sorry, 1,600 participants for community QC. I'll go into this a little bit later. and then trained a model to predict the community QC ratings, trained a neural network to predict the community QC ratings for the remainder of the participants. Whoops. Okay, so I mentioned that we ran it through QSI prep. I want to talk a little bit about why that might be important. I'm not sure how many pre-processing talks you had earlier in the week, but the lack of standardization in diffusion preprocessing can introduce a lot of bias in the interpretation of your diffusion weighted data. So to sort of canonical examples, one is that when you're trying to correct for head motion, sometimes people forget to rotate the B matrix as well. And this plot on the left from Leemans and Jones shows the bias that you can introduce in some diffusion measures like mean diffusivity. The top is mean diffusivity, fractional anisotropy, the principal eigenvalue of the diffusion tensor, and then the transverse diffusivity. And you can see, if you, as you go to the right on that chart, you introduce more bias in those measures. The second is that sometimes you get an axis flip in your gradient table. And if you don't. Okay, we're back. If you fail to fix the axis flip in the gradient table, you wind up with ODFs like in the bottom left there. So those should be aligned along the callosum forceps posterior there, but in fact they're rotated 90 degrees. And after fixing it, they look nice like you want them to. So I'm not, there's no test on diffusion preprocessing after this. The solution is really to use kind of best practices for DMRI preprocessing. And luckily, someone else has already made a great package that does that called QSI Prep. There are many other packages, but one of our co-authors wrote QSI Prep, so it was easiest to use that one. Okay. Now, once we have this preprocessed imaging, we would like to get some sort of measure of data quality. This is a pediatric and adolescent data set. Sometimes kids move a lot in the scanner. We'd like to know when they do. So we first recruited 6 DMRI experts to rate a subset of these participants on a 1-to-5 scale. They used this website that I built called DMRI Prep Viewer. And I think I might actually skip the demonstration of DMRI Prep Viewer, but anyway, they rated these subjects on a 1-to-5 scale. And the nice thing is that these experts agree with QSI Prep's automated QC metrics. So what I've shown here is the scatter plot of expert QC rating on mean expert QC rating on the y-axis, along with maximum relative translation, the number of outlier slices, and here the raw neighbor correlation. And far on the right there, you see kind of a histogram of the QC rating. See, it's very sort of bimodal. And not only do experts agree with QSI Prep, they kind of agree with each other. Here's the measures of ICC. You see, I anonymized the rater names, so they all have these kind of silly, like odd tortoise and gloomy, jerky names, but they tend to agree with each other. So great. Hopefully I've convinced you that we have like a nice expert QC measure for a subset. But if I asked my expert raters to rate, the entire 2100 participant data set, it would have taken a while. So that's kind of prohibitive on the full data set. And not only that, we're going to need future releases of HBN. So we'd like not to have to spin up this whole expert rating architecture every time. So the solution there is to use, our solution at least, is to use community scientists. So we built this website called Fiber. You can go to Fiber and swipe left or right. In fact, I'm already logged in. If I just go here, I can get to fiber and play a little bit. And so, this is kind of, if you played fiber, you would have done a tutorial and you'd know, well, this is kind of an okay brain, I'd pass it. This is an okay brain, I'd pass it. This one's fine. I'd like to get to a bad one. There was some banding there, but I passed it anyway. Oh, that's bad. Let's fail that. Okay, so you see, it's kind of like Tinder. You swipe really quick. You can get through a lot of ratings at once. So the idea here is it's not as involved as the expert rating. It's just a quick binary pass fail, but you can do it. You get a lot of volume. So we had 374 community raters provide about 500,000 ratings for a mean of about 200 ratings per subject. And for those raters who rated above a certain number of subjects, we promised them that we would include them as a consortium co-author on the paper. So we got 133 new co-authors as well. So what do we do with these scores? Well, naively, you might think you could just take the mean fiber rating, the mean community rating, but it turns out like some people are better than others at rating images. So in fact, we trained an XGBoost algorithm, which you can view as kind of like weighting of the expert rater community raters based on how well they agree with the experts. And if we train that classifier to predict the expert ratings using both the fiber scores and the automated QC metrics from QSI prep, we do a pretty good job. Here I'm showing an area under the receiver operating curve of 0.96. So we do an okay job predicting these expert ratings. Okay. So that's great, but then what do we do for the missing subjects and what do we do for future releases? We also don't want to have to go back to our community raters every time we get new participants. So the challenge here is to, our approach here was to use a neural network to predict the QC ratings from the pre-processed diffusion data itself. And I won't go into the architecture too much here, but it's a 3D convolutional neural network that takes in the diffusion weighted imaging. Okay, here are some loss curves. If you want to talk about deep learning stuff with me, you can do that. We can do that later. But the main takeaway is that our neural network seems to do a pretty good job of predicting the QC scores. So finally, we have QC scores for the entire data set without having exhausted our expert raters and without having to keep going back to our community raters. Great. Okay, so I've just gone through all this rigmarole to provide QC ratings. You might be asking, like, well, why should I care, Adam? Is that important? Should we have done all that in the first place? What can we do to demonstrate the importance of QC? So to demonstrate on one of the sort of openly available phenotypes, something that you don't need a data usage agreement for, we picked age as an open phenotype and noticed correlations between age and QC score here on the left, and also group-wise differences in QC quantiles between the track profiles. I think this is for the SLF left, sorry, the superior longitudinal fasciculus on the left side of the brain. So you can see there are real differences between low quality and high quality data. And if you didn't incorporate QC into your downstream analysis, you would add bias. Particularly in the track profiles, you would find localized track profile differences between your groups if you didn't account for QC. And finally, we trained a model to predict age and varied a QC cutoff at the beginning. And lo and behold, if you make a more and more strenuous QC cutoff, we did a better job of predicting brain age from diffusion track profiles. Okay, who should use this data set? That's a trick question. Everyone should use this data set. In particular, I think it might be a really good use case for a NeuroHackademy project. One, if you have interest in pediatric neuroimaging or adolescent neuroimaging in general, or mental health in general, it's a great data set to use. If you have current or maybe anticipated access to the phenotypical data through a data usage agreement, you can kind of get a head start during the hack week on the imaging data, and then hopefully get access to the phenotypical data later. or you may want to examine some of the openly available phenotypical data, which again is age, sex, handedness, and QC score. Actually, we released the QC scores, all the various different types of QC scores as well, in a large-ish cross-sectional data set. You may also want to benchmark your own pre-processing methods. If you were, looking through the pre-processing slide and that was like really your jam, you could pilot your own pre-processing methods and then use HBNPod2 as kind of a benchmark. Maybe you can do better. And lastly, if you're interested in QC of large neuroimaging data sets, you could look at the openly available QC values and see how they map onto each other, how they map onto phenotypes of interest, and so on and so forth. So I won't read this table to you, but if you go to the link in Slack, you can go to this last slide and see this is the table of HBNPod2 data records and where they're all located on S3. So, we provide you with track profiles, QC scores, streamline counts, all the fun stuff you might want. Am I doing on time? 3 minutes. Great. I have time for questions. Oh, thank you.
So you mentioned that you have trauma data on these subjects via self-report. So I was kind of wondering what that looked like. Do you know what measures you used or how you looked at it?
Yeah, that's a great question. So I think in the first release 1 and release 2, they used the ACE. And then they switched over to NLES, the negative life events scale. And NLES is available for the overwhelming majority of the participants. I think that's the only self-assessment of trauma that's available in HBN. And then also you can look inside of the CASE-ADS questionnaire, and there are specific questions about trauma. I mean, very specific questions about trauma in CASE-ADS. Sorry. No, it's all cross-sectional data. Yep.
Hi. Okay. So I have 2 questions. The first is, if I wanted to take this model of making QC, could I apply it to a different large data set? Like, could I put on ABCD and get like a similar metric? Okay.
Maybe ask the other question.
Okay. Yeah, sure. Okay. And then the other question is, I was wondering if I wanted to include quality as a regressor in one of my models. I know, so I've been working with this data, and the QC scores, like the predicted ones, are kind of very bimodal, kind of centered towards 0 to 1. So they weren't like a well-behaving regressor, like you want something like more normal. So I've been using like the neighbor correlation, since that was like the strongest predictor of QC score. But I was wondering if you were to include a regressor for quality in your models from these outputs, what you would use.
Those are great questions, by the way. Are you reviewing our paper, by the way? So the first answer is no. I wouldn't trust our QC network on a different data set. I do think it'll work on future releases of HBN from the same sites. But we in the paper we did sort of some simulations to try and address generalizability. It doesn't generalize across sites within the HBN, so I don't. Well, it gets complicated. It can generalize across sites in the HBN, but not predictably so. I can't guarantee it. And so I don't expect it to generalize across studies. So that was the first question. And the second one was, sorry, could you remind me what the second one was? Yes. Which quality regressor should you use? That's a great question, and I would try a lot and see what sticks, honestly. So we address this with the reviewers. I think all these QC measures should be viewed as exclusion criteria, not inclusion criteria. For example, the QC metrics are some subjects have very high data quality, but also very abnormal brains. So you need to look at something else. I'm not sure that any one metric will capture, inclusion. And if we view if I answer that question in terms of inclusion exclusion, I think I have to answer the same way in terms of model regressors that you include multiples. I know that's not a very satisfying answer, but it's what I believe.
All right. Thanks, Adam. And we'll move on to Kendrick, I believe.
It's hard. Right? All right. Because I think we're running behind already. Really? Only 10? Test. Okay. Hello, my name is Kendrick. I'm a faculty at the University of Minnesota, but actually, before I get to the talk, you may have seen on the Slack channel a smorgasbord of statistics, Fmri analysis, PCA, I don't know, a bunch of stuff that I have some of these I have slides on even, and other ones we could be a free-flowing discussion. I have a million figures on my computer that I can talk about and discuss whatever you like. I'll be around through Tuesday, so Monday and Tuesday, leaving Wednesday, and feel free to Slack me or just tap on me and ask me random questions. All right. So Ariel asked me to talk about the natural scenes data set, which is an effort that we recently completed. And basically, I'll give you an overview of what's in there and why this data exists and what you might want to do with it. I guess it's a little funny. I mean, NSD also suffers from the sort of reverse problem. Typically in science, we have a question, we design an experiment, we get the data. But here, and especially at this Hack Week or Hackademy, it's sort of the reverse. Given this advent of large data sets out there, the data came first, and then the question in some sense comes after. The natural scenes data set is also like that. This data is intended to be a very generic and general data set that many people might be very interested to look at. The motivation for this from sort of our lab's perspective is a very general one. We want to understand at least how the visual cortex works, so vision. And there are many ways to answer this question. One way is to take a very computational modeling focused approach. And you can think of this as trying to characterize explicitly the computations that your brain and your neurons in different visual areas in your brain are implementing to transform visual input into useful forms. And you can formalize this in explicit models, image computable models, or things that actually run on your computer and implemented in your favorite language. And that's sort of the motivation for building this data set. And in this endeavor, one of the limitations of this is basically we're data limited. We need to sample many stimuli in order to get a better understanding of what all of your neurons are tuned to and things like that. So this data set, really the goal here was to establish a very large or massive data set that hopefully will become a benchmark for this part of neuroscience. And again, to be a general purpose, useful database that's freely available right now that you can download and potentially answer hopefully interesting questions about vision. In designing this, we deliberately tried to make this as good and amazing as possible. Of course, there's always limitations. But just some of the fields out there that hopefully some of you have a sense of, different conferences, different communities of people, visual neuroscience for sure. This data set actually involves a lot of so-called cognition. There's the whole memory component, so that's more maybe cognero. Computational neuroscience, which I've already alluded to, is maybe a separate community of people. Theory even, I'm guessing the intersection of this crowd and theoretical neuroscience is fairly small, but there's a lot of theoretical neuroscience in kind of the traditional computational neuroscience sense. So neural coding, correlations, and sort of theories of why the brain is tuned in the way that it is. And finally, the neuroimaging field, which probably is the most intersecting with the people here. There are many sort of interesting uses of this data set from that angle, I would say. Okay. You've already heard and are aware of many very diverse, large number of publicly available data sets out there, as some of the ones I've listed here. I just want to comment briefly, as a data collector, one has to make hard choices about experimental design, and what type of data might actually be useful for yourself as well as for others, if you're so kind as share the data set. So in designing the natural scenes data set, we spent a while designing it. There were a couple sort of priorities we sort of decided at the start. We wanted to be very big, as I've already alluded to, but specifically in the fashion of getting a lot of data per individual subject, which is very unlike many other data sets. We had 8 subjects, which in my book is actually quite large, but from a very different point of view, it's very small. That's a separate discussion. And right, so we have a moderate number of subjects, but a lot of data per individual subject. And you may have seen this figure. This is a real plot. It's a quantitative plot, plotting just a handful of Fmri focused publicly available data sets out there. The 2 axes are the x-axis is just sheer number of hours of Fmri acquisition per subject. And the y-axis, notice is on the log scale, is the distinct number of distinct subjects in a given effort. And so there's a wide range of choices in this 2-dimensional space. NSD, as you can see, is pretty far to the lower right, but there are other ongoing efforts that blow this out of the water in some sense and are even off the axes here. And you can see, classic other data sets and where they sort of chose to fall on this sort of trade-off between, for a fixed amount of scanning time, for example, where do you want to allocate your resources? So this is sort of an interesting graph that you can kind of stare at. And if you are a data collector, you can sort of think about where you might want to fall in this space. Obviously, we want to be in the upper right, but time and money is limited. All right, so we want a very large data set, sort of a so-called deep data set. We also want a very high SNR. We were able to scan at 7 Tesla, which is obviously a big advantage to kind of get better SNR and or spatial resolution. One thing we did, this was not a unbiased sampling of subjects. We deliberately screened subjects, and we have evidence, and it's in the paper, that this was actually a really good idea if you're trying to maximize, say, SNR. And as a sort of methods-driven lab, we definitely developed some novel methods to sort of cope with this kind of unprecedented scale and style of data set. All right, what was the experiment? I say natural scenes, we're talking about visual natural scenes. So there's no auditory stimulation. This, of course, is a choice. There's a rich, you could show movies, of course, with narratives, but here we're talking about single images, and this is literally what the experiment is. And if you know about vision, this is actually a very interesting and rich data set. And if you're not, maybe you think this is boring, but that everyone has their own research questions. So your job is to fixate the small dot here, which is hard. And again, for those who understand vision, fixation is really important. Your whole visual system is basically relative to exactly where your fovea is. So your job is to fixate. And as each of these images come up, you're making a judgment as to whether you've seen that image before at any point in this experiment. And that means either in this run, in this scan session, a week ago, 6 months ago. It's very challenging, but it keeps you on your feet. It's sort of interesting that you're hopefully not bored out of your mind. And interestingly, it is a memory task in a sense. And there are certainly many memory related questions you can do with this data set. So we're showing thousands of these rich, complex natural scenes to subjects as they maintain central fixation and sort of pay attention to the images and do this memory task on them. And this is literally the timing. So for vision, it's very slow, but fMRI is necessarily slow if you know what the signal source is, the blood. And the timing is 3 seconds on, 1 second off. That's the core of the experiment. And if you're into cognitive neuroscience or visual neuroscience, you hopefully have already some ideas of all the many things you could do with this data set, assuming if it's good quality, studying different visual dimensions, building models that can predict brain activity, maybe directly training neural networks to reproduce brain activity, brain optimized neural networks, so to speak. The list goes on and on. If you like mapping out areas in the brain, this could be a great data set for that. It is a multimodal data set, which I'll talk about in a second. So maybe there's some integration across fMRI and say diffusion you could do, so on and so forth. And maybe develop methods, as Adam mentioned, with nice, large benchmark data sets. You can do things like take that data and develop better methods and compare different methods on. And that's something that my lab likes to do. Okay. Yeah, NSD is already freely available, and it looks like it's being picked up by the community. I guess the first people to pick it up are largely the computational and visual neuroscience communities. And I've listed some recent preprints and papers already that have used NSD on the data manual. Okay, let me speed along. Hopefully, yeah, okay, oh, I'm doing okay. This is just one of the figures from the paper. There's a citation here somewhere. It came out earlier this year, I think. There's a lot going on. I'm just giving you a broad overview. There's localizers, as is classically done. So these are Fmri experiments of their own. There's actually a substantive amount of resting state on these subjects. We get a variety of structural measures. Many of them are very standard, as you know, T1 weighted, T2 weighted structural images. There's physiological data collected in a large subset of the scan sessions. Eye tracking was collected, but only in a very few, and it's very hard, at least in our 7T scanner, to get very good quality eye tracking. So I caution that it may or may not be super usable. Of course, you could develop methods to make it usable if you're of that mindset. Anyway, so there's a lot of data. So I wanted to give you some numbers. That's just sort of a qualitative overview of let's look at a quantitative assessment of what's in this data set. As I mentioned, it's all immediately accessible. There's no like paywall or anything. These are all directly downloadable from S3 right now. And the data are in both raw form, so BIDS format, as you've heard about exhaustively already, as well as the pre-processing that we've done using what we think might be good methods. And we actually develop new methods in a sense to sort of optimize this data set. In terms of what's in it, there are, where are my numbers? So there's both functional and anatomical data. We've already said that. Yeah, some of the MRI details, if any of you are into sort of MRI physics and whole sequence choices, we can talk about that. It's a 1.8 millimeter spatial resolution whole brain. So this is fairly good. We definitely didn't push it sub-millimeter, which would cause a huge hit in SNR. So we've sort of chosen hopefully a sweet spot. And yeah, we used head cases to help mitigate head motion. Numbers, right? 8 subjects. Half the subjects participated in all 40 scan sessions. So that's a visit to the scanner for 40 weeks, basically. And within a scan session, they're basically doing this strenuous experiment for an hour. So about 30, so per subject, 30 to 40 hours of this experiment that you just saw, not including these other measures that we did acquire on these subjects. We put a lot of effort into QC, as we just heard about from Adam, but in a very different type of data set in terms of looking at MRI quality, head motion, the usual culprits here. And we developed, which I'll talk about a little bit, of novel methods to try to estimate as accurately as possible every trial, the evoked response in the brain. So we call those single trial bold response estimates. And you'll hear about that in a second. And the pre-processing, again, you can analyze raw data, but we did apply pre-processing using some methods and ideas that we developed in some prior studies. And that's, again, talked about in the data paper. Okay, I think I'm doing okay on time. I just want to sort of bird's eye view, tell you a little bit about the methods. Ariel is on one of these. Oh, there he is. So one of the things we worried about was the resolution. So we didn't push the acquisition resolution that much, but we do care about spatial resolution. And so there was some motivation to actually upsample the data. Now that's a very controversial potentially or tricky topic. I'm happy to talk about it in detail. In terms of the analysis of the data, so remember these images are coming every 4 seconds, which is fairly rapid from an Fmri point of view. So this poses some challenges in terms of deconvolving the overlapping hemodynamic response across trials. And so prior to releasing the data, so we had to develop some efficient statistical methods. Fractional ridge regression is what we called it. This was done with Ariel. You can check out that paper. And that was incorporated into number 3 here, which is sort of an integrated algorithmic approach to GLM analysis of the time series data. And this has been codified into its own little tool, which is on GitHub. You can try this out and or discuss it with me if you're curious. We call this tool GLM Single. Yeah, and that's actually one of the breakout sessions that I suggested we could do next week. Okay, let me skip this slide. I'm more than happy to talk about Fmri pre-processing and how we think about it and lots of fun stuff like that. And let me just end. So that's just, again, bird's eye view. There's a lot more detail. One source of information is this. This is really just like a site that points to other things, naturalscenesdataset.org. The main source of information, I would say, is the actual written paper, and there's a lot in there. If you, especially in the supplementary information, we try to be meticulous and comprehensive and describing what the data are and all the things that we did in the pre-processing of it. So I encourage you to at least browse it if you want to try this data set out. In addition, all the sort of nitty-gritty, gory details of files and exactly what the files are and all that stuff is on the so-called NSD data manual. These are hyperlinks, by the way. So those are your two main sources. There's also a video where I talk you through the files and show you what's what and sort of give you some conceptual overview. It's about an hour. It's just on YouTube. You can watch that. And there's also a tiny little utility GitHub repo to help transform data in different spaces that we developed. Okay. Wow, that was fast. Oh, right. There's one more slide. Sort of circling back to the topic of here's the data, now what do you do with it? And these are, I think, important questions that the field has to grapple with. You know, in what paradigms does it make sense to develop like a general use data set, and then versus other types of studies where you're really tackling one question and you really need to optimize one, perhaps not very general experiment to answer that one question. So just thinking broadly in terms of why should you care about this data set? Well, it depends on what you care about in terms of understanding the brain, assuming that we all are interested in understanding the brain, either structurally, as we heard about diffusion measures, and or function. I guess from my point of view, there are some sort of themes that NSD has really hit on. As I very briefly mentioned, this kind of choice of deeply sampling a fairly small number of individuals. I guess that comes from my lineage and visual electrophysiology, that kind of field, where this is very common due to other reasons. I guess NSD brought in some advances in high field fMRI and ways of processing such data to optimize their quality. I think the primary audience for this data, which may or may not be some of you in the audience, it would be the more computational minded people building, say, deep neural network models of visual activity or other types of models. So I think that community is super happy with this data set and will probably lead to many papers. And from a more neuroimaging perspective, we wanted to include certainly multimodal data sets in resting state even, and different communities gravitate towards to specific certain types of data, but combining and integrating approaches and ways of thinking about the brain across different types of data, I think, is another theme that NSD hopefully will promote in the years to come. Now, being sort of methods minded and thinking about limitations and certainly when we're designing this data set, we of course want it bigger and better and more amazing, but there's always limitations. And I just wanted to mention these, and happy to discuss if this floats your boat. In some sense, from a vision science point of view, there was very little stimulus design. One thing I forgot to mention, these stimuli come with annotations. These were taken from a computer vision or a computer image database. So these are used, it's from the Microsoft Common Objects and Contexts, or COCO image database. So these already have like segmentations and certain objects are labeled. Of course, these are computer vision people labeling them, so they may not be necessarily neurally relevant, but they exist. You could, of course, annotate the images yourself. But anyway, so there was some choice along those lines, but these are definitely not sort of controlled, gradings and dots. And that's a large choice. Anyway, so hopefully the data are still useful, and presumably all stimuli tell us something. Right, the scale. So it's a big data set. You need big computers, you need cloud compute, you need to know how to program. Certainly that's not a big issue for the people in this audience. Yeah, and this is a fMRI data set. And as we all know, fMRI has strengths and limitations. Maybe in the future, we will also collect, maybe not in the same subjects, but use this type of experiment, these stimuli, this kind of memory paradigm in other modalities like IEEG. Actually, I should mention Dora and I are working on that with design choices, of course, given that these are epileptic patients, or EEG or MEG or other behavioral measures for these images and so on. And so we have to be careful to think about the limitations of this data set at the same time thinking about what we can do with the data set, sort of a push-pull judgment here. Okay, that's basically it. And I certainly don't have to tell this audience about, given the existence of the data set, there's many potentially juicy things one can do it if one has computational chops or machine learning chops or neural network, deep neural network chops. And so, yeah, I will just leave that there. And maybe one last cautionary tale. While yes, we can do all these things with the data, but in the end, it should be driven, I would say, by actual theoretical neuroscience questions. And there's a wide variety of them out there. And so I don't want to bias you or say you should only do this question. Hopefully there are many questions you can think of that are interesting to pursue now that this data set exists. That's all I had. And right, and just a reminder, it looks like there was a lot of interest on the Slack in these little breakouts. There were too many topics, so we'll try to be efficient given this schedule, as you can see here. Thank you.
Yeah, we can take some questions too. I'll run.
Okay. Hi, thanks for the great talk. I was just wondering about the use of possible BIDS apps, this data set. Let's say Fmri prep. I think in most use Fmri prep on 3T scan data. And I'm wondering if things would change with 7T. Yeah, I've seen things being trained on 3 Tesla data sets. So I'm wondering if those would be applied on this.
Yeah, sure. Just briefly, the question was about BIDS apps and maybe using Fmri prep on this data set. Yeah, it's certainly doable. I mean, the data are in valid BIDS compliant, hopefully, format. So yeah, we did our own so-called pre-processing, and certainly if your motivation was to compare, sure, there's a million interesting methodological comparisons, some more interesting than others, but so yeah, you could run the data through Fmri prep if you like. And I think you also asked, are there like gotchas to watch out for? Yeah, 7T, I mean, the spatial resolution is a little higher. I mean, in other projects in the lab, we deal with like 0.8 millimeter data, which is potentially qualitatively different. Here it's 1.8 millimeters, so maybe not terribly qualitatively different, but even at that spatial scale, you have to think a little bit about the cortical thickness and, you know, depth dependent or layer dependent Fmri at 1.8. Distortions are larger. So field maps are probably especially important. This data set, since it's many sessions on the same individual, the importance of like within subject consistency is very important. So making sure the brain is lined up as well as possible across 40 scan sessions. So that's something to watch out for. But in general, yeah, the pre-processing issues are not fundamentally different. There's motion, there's distortion, there's registration, which is all the same stuff that happens at 3T.
Oh, one more question up here. Yeah. This far from this, maybe come here, Kendrick. Does it work? Yeah, we can't take. Does it work now? Yeah, we can't really go further than this. Apologies.
Hello, thanks for the talk. I only had 2 quick questions. I was wondering about the signal strength subcortically. Did you look for it in LGN or, for example, Pulvinar? And my second question was regarding the baseline. Did you have a fixation-based baseline? Thanks.
Thanks, great questions. First question was about subcortex. Yeah, I mean, we have other projects looking at this data in the Pulvinar. Funny you should mention. It looks workable. I mean, SNR is always weak in the middle because they're so far from the RF coils. But yeah, as far as I can tell, there's nothing in this data set that says that you shouldn't look in subcortex. Also cerebellum, there's really good signals in the cerebellum if that's something, if that's a brain structure of interest to you. Yeah, and the 1.8 millimeter resolution, I guess, should also be helpful for subcortex. It's relatively high, I guess. The other question was, oh, baseline, right? Yeah, so sorry, in the experiment, there are blank trials. So images are usually 3 seconds on, 1 second gap, but there are occasional like blank trials, which serve as effective baseline. So they're just staring at the dot. And there's some staring at the dot at the beginning and end and of the run. So yes, the implicit baseline is I'm sitting there and waiting for image, I guess, if you want to call that rest, sure. But yes, thanks.
All right, thanks, Kendrick. And we'll move on to the last data set. Oh, okay. I got it.
Okay, this is my Zoom window, so I'm having some technical issues. 1 second. I may actually need to log out of Zoom. Well, this is Zoom. Well, sharing my screen would be about. This is how Zoom is showing up. So I'm gonna. That's going to stop the recording, unfortunately. For those on Zoom, we're having some technical issues. 1 second. Hey, Zoom people, can you hear me? Okay, 1 second. Okay, now Zoom people, are you seeing my slides? Okay. Great. So I'm just going to give a very brief introduction to the Human Connectome project. And before I do this, I just want to state 1 or 2 caveats. First of all, I'm not actually directly involved with the Human Connectome project. I'm not part of that group or anything. I've just worked with the data quite a bit. And in fact, a few years ago, Kendrick and I published a paper on the Human Connectome Project 7 Tesla Retinotopy data set, which is basically a reanalysis of some of the data they collected. And so if you're interested in vision, that's a potential direction we could go with this data set next week. And I'd be happy to talk about that more. So the Human Connectome Project, which is often called the HCP, is a foundational data set in human neuroscience. It was started in 2009 as a National Institute of Health Blueprint Grand Challenge with the goal of collecting an unprecedented data set of both functional and structural connectivity in the human brain. It was originally funded through 2 consortia. First of all, the Harvard Massachusetts General Hospital and the University of California, Los Angeles consortia led by PIs Bruce Rosen and Arthur Toga. which primarily focused on developing Human Connectome Project technology. And notably, the Massachusetts General Hospital, which is often called MGH, was the site at which FreeSurfer was developed as well. So if you're a FreeSurfer user, you might recognize MGH as the ending for a lot of their file extensions. Second, the Washington University and University of Minnesota consortia, led by PIs David Van Essen and Kamil Ugerbil, focused on collecting a massive data set from young, healthy adults. And that's most of what I'm going to be talking about because it's the part of the Human Connectome project that I'm most familiar with. The NIH additionally has funded follow-up projects, including, for example, the Lifespan Connectome, which tracks adults of all ages, the Disease Connectome, which tracks adults with clinical diagnoses. There's a few others. I know there's a psychosis one, there's a low vision one. This is not an exhaustive list. Importantly, all data from the Human Connectome project are publicly available through a single coordination facility at humanconnectomeproject.org. Sorry, humanconnectome.org. And if you go to their website, db.humanconnectome.org in particular, that's where you can download a lot of this data. Like I said, I'm going to focus on the young adult Human Connectome project for many reasons. Mainly, I'm most familiar with it, but also it's often the most useful to students in this course, partly because it has a lot of really great structural data that's very easy to access. That said, I encourage you to check out the other Human Connectome project resources as well, especially if you're interested in aging or disease. So the Young Adult Human Connectome project is a massive data set of approximately 1,200 subjects. You're seeing the right hemispheres of some of these subjects morphing from one subject to the other here. Yeah, approximately 1,200 subjects, all aged 22 through 35. The data set contains a wide variety of neuroimaging data, including high quality MRI scans, diffusion weighted imaging, both task evoked and resting state functional MRI scans. And those tasks include some working memory tasks. For some subjects, it includes vision tasks and a variety of others. It includes 70 MRI for about 200 of the subjects. It includes various behavioral data for all participants. It includes genetic family data for about 300 twin pairs. So if you're interested in twins or inheritance or genetics, this is a great data set to use for that. Unfortunately, it's not a great data set to use next week on a hack project for that, just because the restricted data in the Human Connectome project, which is the data set that includes all of the twins, takes a little while to get. You have to sign some forms. The rest of the data set is pretty easy to get. It takes about 5 minutes to register. Let's see. It includes both task evoked and resting state MEG scans for about 100 subjects. And these data all exist both in raw form as well as in various stages of pre-processing. The pre-processing was actually done with a standard pipeline, or a now standard pipeline called the Human Connectome Project Pipelines, or HCP pipelines. That's available on GitHub. And it's also part of Fmri prep. So if you like the Human Connectome project's way of analyzing subjects or analyzing data, Fmri prep supports that as well. These pipelines are actually based on FreeSurfer's pipelines, and they add some additional tools like the multimodal surface matching for inter-subject alignments. FreeSurfer has a particular way of doing structural based alignments between subjects. And multimodal surface matching takes into account some other data, such as some of the resting state data, in order to better align subjects. So one of the best things about the Human Connectome project dataset is how easy it is to gain access to the data. All Human Connectome project data are available through the HCP coordination website that I mentioned earlier. However, they're also available on Amazon's S3 service. Querying and downloading data from the Human Connectome project is as easy as using the AWS S3 command line tool, which I'm going to show in this demo window on the right. Note that you have to obtain Amazon credentials to do this, and you can do that, like I said, through the Human Connectome project coordination facility. It just takes a few minutes to register, and it's totally free. You don't have to pay anything. So the data live on an S3 bucket called HCP Open Access/HCP, which is being queried using the AWS S3 LS command here. This results in a long list of directories whose names are 6-digit numbers. These are actually subject IDs for all the Human Connectome project subjects. If one looks at one of these subjects directories, here we'll look at 111.312. Inside of each of these directories are a number of other directories, including one that we're just going to look at, for example, called T1W for T1 weighted. And back. Okay. I think the batteries are low, but we've just got a few minutes left, so I'll see if I can make it. Sorry, I was saying, so if we look inside this T1 weighted directory, we can see a bunch of subdirectories for diffusion data, for native structural data. There's an FS average data directory where we have inter-subject alignment data, and then a bunch of files, including nifty files like T1 weighted and T2 star weighted images. Beyond S3, there are also a bunch of existing tools for interacting with the Human Connectome project data. The first of these I want to mention is the Human Connectome project's official tool called Workbench. Screenshots of my back. Okay, 1 second. I'm going to replace the batteries. It'll just take maybe 30 seconds. Can you hear me on Zoom? Okay. Right, sorry about that. So Workbench is an open source C++ program that is somewhat similar to FreeSurfer's Freeview program. Unlike Freeview, Workbench is made to work with the Human Connectome project data. It's capable of understanding all the file formats and data types that the Human Connectome project uses. Another tool primarily for managing and organizing the Human Connectome project structural data is an open source Python library that I maintain called NeuroPython. NeuroPython is designed to work well interactively in Jupyter, and I'm going to show a little demo of it here on the right. So the demo starts by importing the library in NeuroPython, aliasing it NY. Then we're going to request a list of Human Connectome project subjects, which will come back as basically a big tuple. You can see those there. We're now going to request a subject from NeuroPython with the HCP_subject function. And this is going to give us back an object of type subject, which we can see there. Then this subject can then be queried for data such as the hemisphere's white surface. So here we're pulling up the left hemisphere's white surface. That's represented as a mesh object. Similarly, we can get the right hemisphere's peel surface here. It also organizes properties of the cortex, such as gray matter thickness, which we'll see here in just a second. That's represented as an array of thickness measurements, one per vertex on the cortical surface. And finally, it supports various graphical operations, such as plotting those thickness values on a subject's inflated hemisphere. So here we're just going to see an example of the cortex plot function, where we're plotting the left hemisphere, the inflated surface, and we're going to color it according to thickness with the color map hot, which is the same as the hot color map built into Matplotlib. And that looks like this. What it results in is a fully interactive 3D model in your Jupyter notebook that you can interact with and save screenshots of and such. Overall, the Human Connectome project is both an incredibly detailed and useful source of data and a powerful set of tools that set a high standard for large public data sets almost a decade ago. Of course, in fact, over a decade ago. Of course, this showcase was only able to briefly touch on the many details of the project. So I'd encourage anyone who's interested to check out these additional resources listed here. They explain the project much more holistically. That said, I'd be happy to work with anyone who's interested in using the Human Connectome project next week in a hack project. It's a very easy data set to get access to and to interact with. So yeah, and I'd be happy to take any questions. Yeah. Yeah. Yeah, so the question is, in the metadata, do you have information like sex and age? I believe sex and age are both in the restricted data set. I'd have to check. Those two might actually be in the behavioral data, but like I said, I need to check. Generally speaking, the useful data is in the useful metadata for the subjects is in the restricted data set. And that usually takes something like 5 days to get access to. That said, I will say that I have access to the restricted data. So if there's some really cool project someone has an idea for, I'm willing to run your analysis and tell you what the results are without giving you access to it. Yeah. Yeah, so that's a great question. For those on Zoom, the question is, what's the kind of overlap between the Human Connectome project and PyBids, and would it be worth trying to put some of it in some of the Human Connectome project into BIDS, or adapting some of the tools to work with BIDS kind of tools? Yeah, especially NeuroPython. So the Human Connectome project is not in BIDS format, at least not as you download it. It's possible someone else has already put it in BIDS format somewhere. I'm not aware of it though, and it's quite a bit of data, so I would be surprised if they just duplicated it all, I guess. But that said, typically when you run the HCP pipelines analyses, those would go in a derivatives folder in BIDS that would just be the HCP pipelines derivatives folder. So this would not be a very hard task to put it into BIDS. You just need to download the raw structural data, rename that into the BIDS format, and then download all the pre-processed data they have and put that into the HCP pipelines derivative folder, I think. My library is somewhat compatible with BIDS, although BIDS is flexible enough that there's not a really nice interface for it just yet. That's kind of something I'm working on. But it's very easy to use with a BIDS directory or with BIDS data in general. Yeah. 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 Okay. So for people on Zoom, the question is basically the Human Connectome project has kept developing the HCP pipelines, but the last release of the HCP was something like 4 years ago. So would it be worthwhile to reanalyze the data set using the most recent version of the pipeline? Probably. I would say probably not. I honestly don't know what they've changed in the last 4 years, but I think if it was something that needed to be reanalyzed, they would probably have told us and reanalyzed it. They already did that once because of a bug they found where they re-released a bunch of the data. So I think that they're probably not holding out on something great that they've added. That's just kind of a guess though, based on their behavior, not based on my knowledge of the code though. Any other questions? All right. Well, thanks everyone. I think this concludes the resource showcase. So I think now we have a coffee break, and then we will reconvene for the review sessions. 20 minutes? Yeah, sure. 20 minutes. I'm not sure what time it is right now. Okay, so we'll come back and we'll start again at I guess 3:35 after a 20-minute coffee break. And I'll be in this room, and Ariel will be in Alder 103. And yeah, we'll be talking about whatever you'll want to talk about.